Hi, hello, I'm Sandhya. I'm your botany lecturer. In this video, in this lecture, we are going to study about the sexual reproduction in flowering plants. As we have discussed earlier, sexual reproduction. What's the definition of sexual reproduction? What are the events which are going to take place in sexual reproduction? So once again, I'm going to repeat full pressure. Okay, so sexual reproduction means when two parents or opposite sexes participate in sexual reproduction and which involve in the formation and fusion of two gametes, fusion of two gametes is called as sexual reproduction. Here an offspring, the daughter cells are formed which are not identical with that of the parent cell. Okay, so here the fusion of the gametes takes place, that is sexual reproduction. And here sexual reproduction in flowering plants. So in flowering plants, the flowers are there, and this flower is the reproductive structure. The flower is the reproductive structure. Why it is called as a reproductive structure? Because the male and female reproductive structures are enclosed in the flower. Then this is the general diagram of a flower, the longitudinal section of flower. So here the stalk of the flower is called as pedicel. Then thalamus is there. On thalamus, the four whorls are arranged. The first whorl is the calyx or sepals. Then the petals, that is corolla. These are non-essential parts. These are non-essential parts which are mainly useful for, the sepals are useful for protection in bud condition, protection of the flower in bud condition. The petals are useful for attracting the insects for pollination, insect pollination. So these are non-essential organs. The main essential organs, because of this only the flower is called as reproductive unit, is possessing the male sex organ and female sex organ. If both are present, they are called as bisexual flowers or bisexual flowers. If only androecium is present, staminate flowers. If only pistil is present, pistillate flowers, like that. So here, if you take the androecium or the male sex organ, it is generally called as stamen. So this is the stamen. The stamen is having the stalk, filament, and at the tip of the filament, anther is present. So anther is the main portion which is having the male gametophytes, or male which is going to produce the male gametes. Then female reproductive structure is nothing but it's also called as pistil carpal gynoecium. It is a female sex organ, female reproductive structure. And coming on to the parts of pistil, the base of the pistil is ovary. Then the stalk, the tube is called as a style. The tip is called as stigma, where the stigma is the place where it is going to take up the pollen grains and the germination of the pollen grains takes place. Then inside the ovary, the ovules are present, which after fertilization, the ovules are going to develop into seeds and mother, and this ovary is going to develop into fruit after fertilization. Okay, this is a general longitudinal structure of flower. So in this video, in this lecture, we are going to study about the male gametophyte, that is stamen, microsporangium, and pollen grains. Everything about the male gamete, about the male gametophyte. The pollen grain. The pollen grain is the male gametophyte, and that pollen grain is produced in the anther. The anther is present in the stamen. Okay, so we are going to study about the microsporogenesis. So in this video, we are going to study about the mainly microsporogenesis. Genesis, formation of microspores. Microspores are nothing but they are the male gametes, madam. They are nothing but the male gametes. So microsporogenesis. The male reproductive structure is nothing but the androecium or stamen. It is having a filament and the anther. Okay, at the tip of the filament, anther is present. So this anther, a typical angiospermic anther is bilobed. It is majorly bilobed. Okay, and each lobe is having two theca. It is having two theca. Okay, so it is dithecous. Majority of the angiosperms are dithecous, but in hibiscus it is monothecous. Means only one lobe is present. Only one lobe is present, and it is not bilobed. It's only one theca is present. It is monothecous. So here a typical angiospermic anther is dithecous. Dithecous means it is bilobed, and each lobe is having two pollen sacs. Okay, two pollen sacs. Okay, this is about the dithecous anthers. the generally, if we see the transfer section of anther, it will be like this. This is dithecous anther. It is tetragonal structure. This is tetragonal structure, and it is having 4 microsporangia. So and these are the stomium where the dehiscence of this particular anther takes place and all the pollen grains will be released. So in detail we are going to study about this transfer section of the pollen grain. Transfer section of anther. So this is the transfer section of anther. This is the transfer section of anther. Longitudinal section means like this. Transfer section means like this. Okay. So if we see the transfer section of anther, it will be like this. The anther is covered by 4 wall layers. Okay, the anther, if you see the transfer section, the typical microsporangium, it appears, it is somewhat like this tetragonal structure, and this particular anther is covered by 4 layers. Okay, 4 wall layers. The first one is nothing but this is epidermis, the innermost endothecium, this is the middle layer, this is the tapetum. Inside this is the sporogenous tissue, which will be bearing the microsporangium, my pollen mother cells, microspore mother cells, microsporangium, pollen mother cells will be present inside the sporogenous tissue. So this anther is covered by 4 wall layers: epidermis, endothecium, middle layer and tapetum. The first 3 layers, epidermis, endothecium and middle layer, these are useful mainly for the protection and for the dehiscence of the anther, to release the dehiscence, in the sense release the pollen grains. Here the pollen grains will be formed now, so they must be released. So here these wall layers are helpful for the release of those pollen grains by the dehiscence. Then coming on to the, if you talk about endothecium is going to lose water, right, and it is going to contract. The cells of the endothecium, it is going to lose water, and it is going to contract, and dehiscence occurs. That loose portion, is called as the stomium. Here the stomium will be present. That is the portion where the pollen grains will be released. It will be cut and the pollen grains will be released after maturation. Okay, so if we see the detailed structure, the epidermis is a single cell thick, like this, single cell thick. Then endothecium will be there. Then the middle layer. Then the tapetum. Then the sporogenous tissue. Okay, This is epidermis. This is the endothelium. This is the middle layer. Tapetum. This is the spore mother cells or microsporangium. Okay, so mother cells. Okay, this is about the wall layers. Coming on to the tapetum. This tapetum, it is a nutritive tissue. The three layers, the epidermis, endothelium and middle layer, are useful for protection and resistance of the anther for release of pollen grains. But the tapetum is a nutritive tissue. The tapetum is a nutritive tissue. It nourishes the developing pollen grains. This tapetum, it is the innermost layer and it is nearer to the sporogenous tissue. So it is going to nourish the developing pollen grains inside the microsporangium. And this particular tapetum cells are having dense cytoplasm with number of nuclei, more than one nucleus. So this is the tissue. This is the tissue which is having the microsporangium. Okay, this is about the structure and everything. Now we are going to see how the pollen grains are formed. Means the formation of microspores. That process is called as microsporogenesis. Microsporogenesis means genesis of the microspores. Formation of microspores. Microspores are nothing but the male gametes. Okay, so how they are formed? First stamen. Okay, the stamen is having the anther. Okay, the anther is having the microsporangium, isn't it? The microsporangium. The microsporangium is nothing but it is having the microspore mother cells. Microspore mother cells, or they are also called as pollen mother cells. They undergo the reduction division or meiosis. This is nothing but the reduction division. And they produce the microspores. Okay, so these microspores, from one pollen mother cell, 4 microspores are produced. 4 microspores are produced. These microspores are nothing but the pollen grains actually. Pollen grains, or you can say male gametophyte. Okay, why from 1 4 are formed? Because these microspore mother cells are undergoing meiosis. When the cells are undergoing meiosis, from 1 cell 4 will be formed, haploid cells. So here from 1 4 will be formed. So these microspores or pollen grains, they are arranged in a cluster and they form like a tetrad anamata. Okay, they form like a tetrad. Like this. Like this. This is called as pollen tetrad or microspore tetrad. Why tetrad? 4 are present, that's why. Okay, so this is pollen tetrad, microspore tetrad. Then from this tetrad, then they will be released. They will be forming into a pollen grain. Like this. This is the pollen grain. They will get dissociated and they lose water, they get dehydrated and they form into a pollen grain. So the pollen grain is the male gametophyte. And you know, so if we observe this particular pollen grain under microscope, you will be amazed to see the variety of microscopes, their structure, their shape, their size. Different species of plants will be having different varieties of microspores and different shapes, different size, and the design will be different. You will be totally amazed. That's a wonder of the nature. Then so these are the pollen grains. From tetrad will be formed, they get dissociated and form into one pollen grain. This pollen grain is somewhat spherical and it is covered by two layers. The outer wall layer which is called as exine. Okay, it is exine. It's somewhat thick and it is an outer thick wall. It is called as exine and it is made up of sporopollenin. Okay, some special characteristics are there for the sporopollenin. The exine is made up of sporopollenin. It is the most resistive material matter. It can withstand high temperatures, acids, bases. The lava also it can withstand. So it is the most resistive material known till now. Okay, it can withstand high temperatures, strong acids, alkalis. It's like it can resist environment. That's why these pollen grains can be formed as a fossil source. They can be formed as fossils also because the outermost layer of pollen grain is made up of sporopollenin. Sporopollenin is an organic material. Then it is having small apertures in somewhere here and here like this, and those apertures are called as germ pores. Those apertures are called as germ pores. At that portion, the exine is not there. Then this is the outermost thick layer. Okay. Then the innermost thin layer, the inner thin layer is called as intine. Okay. So the pollen grain is covered with two layers. Outermost layer exine, innermost layer is called as intine. This intine is made up of cellulose and pectin. Cellulose and pectin. The composition of intine. Then when this particular pollen grain gets matured, it develops. What happens? It will be having two cells. Okay. The larger one is called as vegetative cell. The smaller one is called as generative cell. Like this. Okay. So the vegetative cell is bigger and it has abundant food reserve because these can be fossilized also, and they are going to survive in high temperatures, and they can withstand also in extreme conditions also they can survive. So how they are surviving? Because this vegetative cell is having abundant food reserves is present, and then generative cell is small, and this generative cell is going to float in the cytoplasm of the vegetative cell. And in 60% of the angiosperms, the pollen grains are going to shed at this stage. Means at this stage, the dehiscence of the anther occurs in the stomium. Then the release of the pollen grains takes place. So those pollen grains are at this two-cell stage. Where the pollen grains have vegetative cell and generative cell. Okay, vegetative cell and generative cell. In 60% of the angiosperms. In rest of the angiosperms, what happens? 3-cell stage, the pollen grains are released. Means what happens here? The generative cell is there. The generative cell is going to divide by mitosis and form into 2 male gametes. So in the 3-cell stage, in some angiosperms takes place. The dehiscence of the anther takes place. Then what happens? Now this particular pollen grains This particular pollen grains are very much useful for the continuity of the generation of the plants. They are the male gametophytes. They possess the male gametes like that. But in human beings, they are going to cause some allergies, some small complications like allergies, bronchial afflictions like that. In some individuals, in some cases, what happens, it can lead to chronic respiratory disorders also, like asthma, bronchitis, etc. So they may cause the allergies also. Then these pollen grains are rich in nutrients. They are very much rich in nutrients, and they can be stored as pollen tablets and be taken as food supplements. They can be taken as food supplements because of the presence of abundant nutrients. In Western countries, large number of tablets and syrups are prepared using these pollen grains. Pollen products are prepared. Like tablets and syrups in Western countries. They are nothing but used as food supplements. They are going to increase the viability of the athletes and racing horses. So that's why they are going to give as a food supplements to the athletes and racing horses. Then coming on to the sexual reproduction, what happens at this 2-cell stage? The pollen grains are released. Then what is the destination of this pollen grain? It has to fall onto the stigma, and it has to fall on the stigma in such a way that it should be viable. It should take part in the reproduction process. Okay, it should have that viability to take part in the reproduction. It should not lose its viability. So in that period only, it should fall onto the stigma and germinate. So once they are shed, pollen grains have to land on the stigma before they lose the viability if they want to bring about fertilization. So if they want to take part in fertilization, they have to have the viability, and they have to fall on the stigma. Okay, so this is about the pollen grain. These pollen grains should have the viability when they fall on the stigma for the fertilization. This viability period varies from species to species, and different species are having different viability. If we take an example, in cereals, in rice and wheat, the viability is only up to 30 minutes. So when the dehiscence of the anther occurs, when the release of pollen grains occurs, within 30 minutes they must fall onto the stigma of the another flower. Otherwise, they will not germinate. They will lose their viability. They will not germinate. They will not take part in fertilization. So within 30 minutes, they should reach the destination. That is, the stigma fall onto them, and then germination should occur. Otherwise, they will lose their viability. Whereas in case of Rosaceae members, Leguminosae members, and Solanaceae members, they have the viability of the pollen grains is up to 1 year. So within 1 year, if they fall into the stigma and germinate, they can cause the fertilization. The viability will be there for months together. Then these particular pollen grains can be stored as a pollen banks in liquid nitrogen. -196 degrees centigrade. They can be stored for several years, and such type of pollen grains are called as pollen banks. Similar to that of seed banks. Hence, they will not lose their viability anamata when we store them in a liquid nitrogen at -196 degrees centigrade. So the viability also depends upon the temperature and the environmental factors. So if you take for example rice and wheat, they are going to lose within 30 minutes. So if you store them and use it, then it is useful. So the pollen banks are very much useful as seed banks. Seed banks are useful for crop breeding programs. Then in Rosaceae members and Leguminosae members and Solanaceae members, the pollen grains are viable for up to months together. So if you want to store for more and more years, you can store them in liquid nitrogen at -196 degrees centigrade as pollen banks. Then they can take part in the fertilization. Thank you.